Welcome to The Explainer. Today, we're unpacking a truly classic biotech chapter, but we're going to look at it just a little differently. We're going to treat biologists like microscopic detectives. Think about it. Every single day, researchers are tasked with solving incredibly complex mysteries. They're investigating biological crimes, like disease, or deciphering the very blueprint of life itself. But their prime suspects, the evidence, the crime scenes, they are completely invisible to the naked eye. So how do they do it? Well, they use an incredibly sophisticated toolkit, so get ready because we're going on an engaging tour of the tools and technologies that make modern biotechnology even possible. So that brings us to the core problem of this chapter. How on earth do we study a world we literally can't even see? It's so easy to look at the results of biotechnology, like a new mRNA vaccine or a drought-resistant crop, and kind of think of it as modern magic. But it's not magic at all. Biotechnology is a highly experimental, grounded science. It relies entirely on extremely sophisticated physical and chemical tools to actually uncover evidence. Without these laboratory methods, the field just simply wouldn't exist. We literally have to manipulate the physical properties of molecules, things like light, mass, charge, and density, just to get a tiny glimpse of what's happening at the cellular level. Here is our roadmap for the chapter. 1. Seeing and locating. 2. Separating and sorting. And 3, analyzing and decoding. Let's get right into it. Section 1, seeing and locating, looking beyond the human eye. Okay, let's dive into this history because the sheer speed of this technological leap is just mind-boggling. Back in 1665, the British physicist Robert Hooke designed a simple microscope using a combination of magnifying lenses. He looked at slices of cork, saw this kind of honeycomb-like structure, and actually coined the term cell. That was the foundational glimpse. But fast forward to the 20th century, and our tools allowed us to see not just the room, but the intricate furniture inside. In 1958, Frederick Sanger won his first Nobel Prize for discovering the exact structure of the insulin molecule. And then, less than 20 years later, in 1977, Sanger completely sequenced the genome of a bacteriophage, the very first genome to ever be completely sequenced. Going from looking at a slice of cork to reading the underlying blueprint of life in just a few centuries is wild, and it was entirely driven by the advancement of these laboratory techniques.
Now, you might be thinking, just use a bigger magnifying glass, right? Well, when it comes to optical tools, making an object look bigger through magnification is really only half the battle. If it's just a giant, blown-up blur, it's completely useless to our detective. That's why resolution, which is the ability to keep two closely situated objects distinct from one another, is the true bottleneck in high-end microscopy. To actually visualize things like DNA molecules, tools like electron microscopes have to master both. They require making the invisible absolutely huge while keeping it incredibly sharply distinct.
Section 2, separating and sorting, isolating the microscopic evidence. Okay, so once we can actually see the cellular world, we immediately realize one thing. It is a total mess. A cell is this chaotic soup of proteins, DNA, RNA, and lipids. If we want to study a specific protein, we have to isolate our suspect from the crowd. Our primary sorting tools each exploit entirely different physical properties. First up, centrifugation. Think of this like a microscopic spin cycle pulling immense g-forces. It generates centrifugal force to separate various biomolecules purely based on their densities. Next, we have electrophoresis. This is basically our microscopic racetrack. It separates molecules like DNA, RNA, and proteins based on their charge-to-mass ratio while they're sitting under the influence of an electric field. Finally, chromatography. If electrophoresis is a racetrack, chromatography is more like an obstacle course. It separates components based on how they interact with a mobile phase, like a solvent flowing through a system, and a stationary phase, using principles like adsorption or ion exchange to purify those specific proteins and nucleic acids. Section 3, analyzing and decoding, solving the molecular mystery. All right, our detectives have isolated the exact evidence they need. The next step, the interrogation. We need to decode what this isolated molecule actually is and what it's doing. Let's look at a highly specific cytogenetic technique called FISH, fluorescence in situ hybridization. It is absolutely vital for understanding chromosomal abnormalities. Here is exactly how it works. Step 1, we design a probe. This is a sequence of DNA equipped with a fluorescent molecule, custom-built to be complementary to our target gene. Step 2, we put the target chromosomes on a slide and denature them. Step 3, we also denature our fluorescent probe and add it right into the mix. Step 4, the probe hybridizes, or binds to its complementary site on the target chromosome. Because the probe is fluorescent, it literally lights up our suspect under the microscope, showing us exactly where that specific gene falls on the chromosome. Building on that capability brings us to a real game changer, the DNA microarray. By exposing a DNA chip, which is covered in thousands of single-stranded DNA probes, to a fluorescently labeled sample, we can actually analyze and quantify the expression levels of massive number of genes simultaneously. The readout acts like a brilliant quantitative dashboard. Classically, green data points indicate upregulated genes, while red points flag downregulated ones. This gives biologists a sweeping, highly data-rich view of cellular activity in just a single glance, essentially allowing them to track thousands of suspects at once. Next up is what happens inside the mass spectrometer. When our detectives need to identify totally unknown compounds or deduce exact molecular structures, they turn to ion sorting. A sample is introduced, it's vaporized into gas phase ions, and fired straight through a vacuum. Inside the mass analyzer, these ions are sorted based on their mass-to-charge ratio using electric or magnetic fields. The detector then picks up these sorted ions and generates a mass spectrum. It's essentially a highly precise chemical fingerprint for our suspects, letting researchers identify exact compounds based entirely on molecular weight alone. To round out our toolkit, let's briefly look at a few other heavy hitters of the modern lab. Biologists constantly use varied physical properties to decode life. For immunology, there is ELISA, a highly sensitive assay used to measure the exact concentration of antigens and antibodies. To decode using light, spectroscopy measures how much light passes through a liquid sample versus how much is absorbed, which reveals exact molecular concentrations. Then we actually use lasers in flow cytometry, passing cells one by one through a beam to rapidly analyze their physical and chemical properties based on how they scatter that light. And finally, the ultimate code-breaking tool, DNA sequencing. From Sanger's early manual methods to today's fully automated next-generation platforms, this determines the exact letter-by-letter nucleotide sequence of our genetic blueprint at truly breathtaking speeds. So going from simple magnifying lenses in the 1600s to passing single cells through lasers and analyzing gas phase ions today, the experimental tools available to biotechnologists are truly the engines of discovery. They take the chaotic, invisible soup of the cell and turn it into measurable, sortable, and readable data. It really makes you wonder, with tools this powerful today, what incredible biological mysteries will we uncover tomorrow? Thanks so much for joining me on this explainer, and keep exploring.